Trying to step up to the plate and prepare some LB agar plates. That is, if I can figure out where all the equipment is and how to work the autoclave here. And so starting out as a postdoc, you think like, oh, I've got my doctorate now, I should be able to be doing all this stuff, and then you realize you have no clue where anything is. And I'm doing some techniques that I haven't done since like undergrad, because in grad school, in our lab, we had like a media maker and we had lab technicians who would do things like make LB agar and these plates, which we can use to grow bacteria in, and we can add antibiotics to select for bacteria that have something that we're interested in, like a plasmid. And so you can actually either get it with like the LB agar mix, where it has like the LB, so the broth, which is actually Lysogeny broth, or then you have the, you can make it from the broth and the agar together. So I'm going to be making some LB and then some of the LB agar plates. And so yeah, so this is more about how you do that and how it all works. LB agar plates, these are really useful for growing bacteria. And when I'm talking about growing bacteria, the reason I'm usually doing it is because I'm trying to select for bacteria that have taken in a plasmid that I want them to take in. And that plasmid has some sort of genetic information, maybe the instructions for making a protein. And it also has an antibiotic resistance gene. If I grow my bacteria in the presence of the antibiotic resistance gene, then only the cells that have taken in the plasmid will be able to grow. And so this allows me to select for those cells. And so I can get them to grow in this food with the antibiotic by putting the antibiotic into the food, into this gel mesh in this agar plate. So one of these plates, and then what's going to happen is the bacteria, they're going to grow. They're going to kind of grow in these little clumps called colonies. And this is really useful because each of those colonies is going to be genetically identical. And well, at least mostly, or at least you hope so, you assume so. Whereas if you were to just say stick the bacteria into a big liquid culture, now you would have all sorts of different like all of those like colonies, so all those individual bacteria, now you would have like an actual mixture. And so there might be ones that actually don't have a mutation or something, that various things like that. And so by plating them, now you're spreading them out, and then they each can take hold and then grow on top of themselves. So you get these colonies that then you can use. So it's really helpful to be able to grow bacteria on these plates. Note that I'm coming at this from like a biochemistry and molecular biology side of things. If you're into like microbiology, you might be using different specialized types of plates and doing things to actually just see what's growing, see what's there, and study the bacteria. But really, I'm just using them as a tool. And so you might also, if you were looking at resources for microbiology, things might be a little different in terms of some of the techniques, maybe using slightly better techniques and being more careful about various things and optimal. But I'm just going to tell you how I do things, and it's worked pretty well, and it's how I was taught and all of that stuff. Okay, so first of all, what is this agar we're talking about? So we have these two things in our LB agar plates. We have the LB, which is the food. It's this like Lysogeny broth, and we'll get into that in a minute, but this is basically just your bacteria food. And it's a pretty basic generic food. There are also different types of media that we can use for different things. Sometimes you might want to have plates that are going to allow for screening in addition to selection. So screening is you're saying, okay, show me something. Selection is saying like, okay, only let things grow if they have some feature. You can also do things like see if, in addition to selecting for antibiotics, you can select for needing something to grow. So if something is oxytrophic for something, you have to add it. If something is autotrophic, you don't. And so this can be used to select for things in a different way, to evolve things, various things. So you can make changes to this basic media formula. But really this LB is the go-to for when we're making these plates. We'll have the LB, and then we'll have our antibiotic that we're going to add to make sure that the bacteria have the plasmid of interest. And as I'll tell you about, we're going to add the antibiotic after we do the autoclaving. So we're going to do this autoclaving. We're going to stick it in this big, really hot, high pressure machine, and it's going to get things sterile and really hot though. And then once it cools down, we'll add our antibiotic. So I'll get into all of this more in a minute. But first off, back to this agar. So why it's so helpful is because it can form this mesh. So don't confuse agar and agarose. So agar is a mix of agarose and agaropectin. So these are these long, like sugary things. And what makes them really great is that they can form these gels. So they kind of like knit them. I like to think of it as like knitting themselves up, because unlike when we're making a gel for, say, like a polyacrylamide gel, and we're actually polymerizing things, like making covalent bonds and actually sticking things together. Here, we're just kind of, everything's just getting knit up into this gel. And so this like mesh that's going to hold liquid inside of it. When we're doing an agarose gel, there we're using agarose, which is this long and mostly consistent and neutral. This makes it really good for separating pieces of DNA by size. And so this agarose gel electrophoresis is a really common technique that we use to separate DNA fragments. When agar, though, it has the agarose, but it also has this thing called agaropectin. This is going to be shorter, and it has more modifications, including charge modifications. So this charge modifications are bad if you want to separate DNA, because when you're separating DNA, taking advantage of the charge of its backbone, and now you have like charged stuff in your gel, yeah, it wouldn't work out too well. But when you have the agaropectin here, it's good that you have the negative charge, because then that's going to help it like hold water, and you don't worry about things like that. And then we're going to add our antibiotic. And so this is what agar is. And we can add this agar to the LB, to our Lysogeny broth. So I had a bit of nostalgia flashback, whatever the term is, to my undergrad when we made LB ourselves, because I was making it here, except here I was making it in an easier way from this actual like bottle of premixed components. So this Lysogeny broth, this is like the Miller formula, which has like a higher salt. So it has like 10 grams per liter of salt. Some of the LB formulas, there's like Lennox, there's Bertani, I don't know, there's some different formulas that have different amounts of salt. But basically the amount you'll put per liter is going to depend on like which formula it is. So you can see that this has 10 of the casein digestpeptones. So this is like cut up, like digested protein peptides from the protein casein. So this is going to give it all the amino acids and stuff. Then it has sodium chloride to give you the salt, and then a yeast extract to give you all like the trace minerals and various things. And so you can see 10, 10, 5, and so 25 grams per liter that I'll make for this. So this is really just a pre-mixed mixture, but you can mix them from scratch. But just a word of caution is that these, especially the tryptone, it's an acquired smell, and it's all so kind of like powdery, and so it can kind of like powder, and then people, if you don't like the smell, then yeah, but it smells kind of like yeasty. I guess that part of that's probably the yeast extract, but yeah, I like the smell of LB. But anyway, so yeah, so in my graduate science lab, we actually had a media maker who actually made the media, and then we didn't have to do this part. But here we do, but it's really fun. Well, I don't know if it's like fun, but it was nice to actually be able to feel like I was doing something productive in the lab, as opposed to just like trying to figure out where the heck everything is. And so today I got to figure out where things were a little, do some actual work, instead of just mostly planning and preparing. Okay, so this is the LB, and we're going to make it from that powder, but we can also make it when you do this way, you just mix the LB in the water to the volume that you want. So I made a liter or I made 500 mils, and then I split it into different bottles. But you can also make LB agar going from the Agar powder. Well, now here you add in an extra 15 grams of agar, so you'll put 40 grams per liter. Oh yeah, and the tryptone is like the same thing as like this casein digest. The percent of agar is going to kind of like depend from formulation to formulation. So this is 15 grams a liter, which is a 1.5%. I've seen everywhere from like 1 to 1.5% of agar. Okay, and so then you just weigh that out, and then you dissolve it. And so basically when you're doing this, it's going to like sediment. You can see kind of here that this agar, it's not gonna like dissolve nicely, but you want to at least mix it. So get it thoroughly mixed so it looks something like this before you actually stick it on. And so you put a stir bar in the bottom and leave that stir bar in when you go to the autoclave, when we get it all sterile. When you autoclave something, you want to make sure that you don't fill the bottles too full. So you want to fill them to like no more than like 2/3 or 3/4 of the way full. Another important thing about when you're autoclaving is that you want to leave the lids on loose. So if you're doing something like a flask, what you can do is you can stick foil over the top. What I like to do is I like to fold over the foil and then fold it over on top. So this way, if there's like a tear in one of the strands of foil or sheets of foil or whatever, then you still have that other additional layer. And then on the bottles, you want to leave them loose. If you don't leave them loose, pressure is going to build up and then this can explode. But you also don't want to leave them like off or too loose because then what's going to happen is that this is going to boil and it can like boil over, which is another reason why you don't want to fill it too full. So in undergrad, they said like, make sure that you don't put them on too tight. And so I was really worried about putting it on too tight, and then I put it on too loose, and then everything spilled over, and then I overcorrected, and then I got a lid stuck on because it was too tight, and then yeah, so just like screw it all the way and then make like one turn or so back around to the left. When you stick something into the autoclave, so the reason why we're leaving the stir bars in here is so that afterwards we're going to add the antibiotic. We want to be able to mix the antibiotic in. And so you can leave the stir bar in there. This way it's going to get sterilized and you don't have to worry about sticking a dirty stir bar into the solution afterwards when you want to mix in your antibiotic. You can also, if you forget, you can just kind of like swirl the antibiotic around if you forgot to put the stir bar in. Okay, so that's the bodily type stuff. As for the autoclaves will look different in different places. So I was really excited because I was going to learn how to use our autoclave, and then our autoclave was broken, and there was like water over the floor. They were like mopping up, but then we were able to use an autoclave on the other side of the building, so I got to learn how to use that side of the building's autoclave. But this is actually the autoclave from from grad school. But when you put things into the autoclave, you want to stick a piece of autoclave tape on them. This tape is going to change color when it heats up in the autoclave. So the autoclave is going to get really, 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 really hot. And so it's going to get really high pressure. This high pressure is going to make it harder to boil. And so things are going to get really, really hot. And so they're going to boil at a higher temperature. And so you're not going to get like as bad of evaporation. And you're able to get really, really hot temperatures to kill things. Okay. So you get these really hot, and you also find that there are different cycles that you can put things on depending on whether you have solids or liquids. If you're putting liquids on, you put it on the liquid cycle, but you can still have solids in there, but they might get a little wet. And so you see when you take things out of the autoclave, there's going to be some like water and that sort of thing. And you can also see that now this autoclave tape, it's changed color, showing you that the autoclave worked. Or at least that it got hot, because this tape is going to change color when it gets hot. Okay, so now you've taken it out of the autoclave and you're using your gloves. So no, I did the autoclave for like 20 minutes, but the whole cycle will then take like an hour because it has to like get up to pressure and then it has to depressurize afterwards. And so, but then you take things out. And now what's going to happen is the next step is going to be to add your antibiotics and then to pour your plates. But do not add your antibiotics right away. Instead, what you want to do is you want to wait till this cools down to about 50 degrees or so, 55 degrees or so. And then you want to add your antibiotic. If you add your antibiotic now, you're going to destroy the antibiotic. In addition to having destroyed the bacteria you wanted, well, now you can't destroy bacteria in the future because you've destroyed your antibiotic. So you want to add the antibiotic after it's cooled down, but you have to be careful about cooling down because then you're going to get this to gellify again. So when we had the solid, it was kind of just like all clumped up and like all weird. And then when we dissolved it, we make this solution. And then the solution, now all these strands are free, they can knit themselves up and form this gel. But it's a reversible gel. So we can melt this back into a solution, and then we can heat it back into a gel or cool it back into a gel. So you can go both ways, which is helpful because this means that if you wait too long and it solidifies, or if you don't have time to pour them right away, what you can actually do is you can just keep the agar solution without the antibiotics in them as just like this, as a gel that you can then heat up. And so you can autoclave it and then you can store this. Alternatively, what you can do is if you want to pour them right away, or at least kind of right away, the best thing to do is to like stick them in a water bath set to 55 degrees. This is really nice because it's going to get it to the temperature like right above where it's going to go back to the gel form. And so especially if you have multiple bottles, and this is going to allow you to keep the bottles from gelifying while you're working with one, you have the others here and stuff too. If you don't have one of these, or if you don't have time, typically, so like what I did today is I just heated it up and then I just let it cool to the temperature where I could comfortably hold it. So, or at least somewhat comfortably hold it. You want to be able to, with a gloved hand, be able to like hold it and pour it and stuff without burning yourself. If you can't do that, it's too hot. If you can't pour it because it's a gel, it's too cold. And so if you want to add the antibiotic at that point, and then work quickly, because once it gets to that gelling point, it's going to gellify quickly. But thankfully we have a little bit of wiggle room because of this thing called thermal hysteresis, which means that it melts at a higher temperature than it gels. This means that we would have to heat it back up to like 90 degrees Celsius in order to get it to dissolve again. But we have to cool it down to about 40 degrees Celsius in order to get it to form a gel. So when we take it out of the autoclave and it's super duper hot, we have all the way until it gets down to about 40 degrees before we have to worry about it gellifying. But then once it gets there, it's going to gellify fairly quickly. And so you start to get these, like, it'll start with like chunks and then it can all go. If this happens before you add the antibiotic, it's okay. What you can do is you can just microwave it, or you can stick it into a water bath. And so additionally, if you have it just stored like this, you can either autoclave it to heat it back up, or you can microwave it. Just stick it in the microwave, give it a couple minutes, swirl it, give it a couple minutes, that sort of thing. But keep a close eye on it. Don't have the lid on there too tight, various things like that. But you can't do that reheating if you've already added the antibiotic, because the reason why we cooled it down before we added the antibiotic was because if we added the antibiotic when it was hot, then it's going to kill the antibiotic. So you can do this, but before you add the antibiotic. But if you already added the antibiotic, then you don't want to reheat it. And so you want to, if you're working with like multiple things, like I was doing three different ones, I worked with one at a time. And so in case they started hardening, then I could have the others. Then I could heat it back up if I needed to. Okay, so that is how you do that. And so how are you actually pouring the plate? So different people have different ways that they like to do this. A key thing is that you want to keep your surface sterile. So what I do is I wipe things down with 70% ethanol. Make sure the ethanol dries and stuff, because then what you're going to do typically is you use a Bunsen burner. So some people, maybe you have like a hood that you can do this in, like a sterile hood, or that sort of thing. But if you don't, you can just work out on the bench, but use a Bunsen burner to keep a flame going, to kind of keep a sterile culture. And then, but you want to make sure that you don't have like the wet ethanol around, or else you can have a big fire hazard. But these plates come in these like stacks and these sleeves, and you can basically just take the stack out, and then you can pour the gels. It's helpful to pour them in stacks of like 5 or so. When you're doing this, you can, so some people like pipette into their plates, but I just pour it. I pour, just kind of gently pour. You don't want to have bubbles, which is, so like if you're shaking or if you're stirring too vigorously when you have your antibiotic, you can introduce bubbles, and then these bubbles are going to make your plate all wonky. And make bacteria grow in weird things around the bubbles. And yeah, you don't want that. So you want to pour, typically I pour, there's like a little lip on the thing, like a little line. Typically what I do is I just like pour gently from like a side, until it, you can see that it's like the liquid's coming together, and it's almost come together, and then it'll all the way come together. And so that's typically how I do my pouring. And then I do this. If you have a bubble, you can either, a couple things you can do. You can take the Bunsen burner flame and kind of like zap the bubble. But you want to be careful you don't over zap. Don't get too zap zealous, because then you can also be killing off the antibiotic. You can also take a sterile pipette tip. So along with the media I sterilized some pipette tips and tips, and then I could use those to kind of just like pop the bubble. When you're doing the pouring the plates, you don't want to just like leave the lids wide open. Instead, just open the lids as little as you have to in order to pour, and do it like that. When you open the bottles, especially if you're opening a bottle and closing a bottle and that sort of thing, take the bottle and just like whip it through the flame, to sterilize the top and stuff. And trying to go in and out of bottles as little as possible. Okay, so make sure that you label everything. So if you have, I was doing 3 different antibiotics, and so I labeled the top of the things, and then afterwards what I do is I color code. So I have like a blue stripe. You can just take a Sharpie off the side of the tubes. It's fun, or the plates, it's fun. So I use blue for kanamycin, red for ampicillin, and green for streptomycin. And then I like doing lines like this too, because then say you have 2 plasmids in there, you have like a can and a strep, then you can put a green line and a blue line. But everybody will have their own system. Okay, and so now I'm gonna let them dry overnight. You can see that they have this like condensation forming. So when you had the bacteria, or when you had the liquid really hot when it was in there. And so now some of that liquid's gonna like evaporate. When it evaporates, it's gonna find this nice cool lid surface. And so it's gonna condense onto there. We wanna try to get all this stuff like evaporate out before we're gonna store these in the fridge. So I'm gonna let them dry out overnight. And what I'm gonna do is I'm going to flip them over so that any condensation that occurs later, it won't fall on the plate. And it will, and I'll keep it in the fridge. You can kind of use the sleeves from what I like to do is I like to use the sleeves from the package that they came in. Sometimes they come in, they have like a separate sleeve inside that makes it really nice, because like when I was opening the package for one of these, I split them. And so now I'm gonna have to just like tape them or something. But yeah, and then keep them in the fridge, and they should last a pretty good time. Make sure there's nothing growing on them, that sort of thing. before you use them. You can also do like a positive control, negative control, check and make sure that they're actually working. Various things like that. Just a couple of quick notes about like the antibiotics. So you want to make, typically what we do is we make like 1,000x antibiotic stock. This is super duper convenient. So you have 1,000 times the working concentration. So if you have to add just like 1,000th of it when you want to use it. So if you have a liter of culture, you'd add a mil of the antibiotic. If you have 100 mils of the culture, then you would add 100 microliters of the antibiotic and that sort of thing. And it makes it really simple. You can see online that they'll have different, you can find like the working stocks recommended for various antibiotics. You want to prepare the antibiotics and filter sterilize them and make aliquots and then store them. I found we have this really cool rotator here and I was just really excited about it. But so I weighed out the antibiotics. I made stocks. I just made like 20 mils and then I'm going to filter sterilize it. So this is like a 0.2 micron filter to filter out anything because I'm not autoclaving these or anything because remember that would kill them, which is why we don't stick them antibiotics in first. But you can sterilize them by filter sterilizing them. If you want to find aliquot sizes, then you could like filter sterilize into something else and then pipette in, but I was just eyeballing about a mil in each of these, and then just stick them in the freezer. And yeah, it's nice to make these aliquots because then you're not having to freeze thaw it all the time. You're not having to worry about contamination going back into and out of the vial or the tube or bottle or whatever it is. Okay, so I think that was basically that. Some people have, like I said, some people have different techniques that you'll see people recommend different techniques. And I'm sure that some of them work a lot better than mine. But this is just how I always do things. And it's worked out for me. And so I hope it'll work out for you too. And happy growing.